Hello, and welcome to Ocean Inverts. I'm sure a lot of you have seen this creature before. It's a sea anemone, and if any of you have watched Finding Nemo, you know that clownfish live in these anemones. Along with being a beautiful home, clownfish use anemones as a defense against predators. Anemones are within the phylum Cnidaria, whose synapomorphy are cnidocytes, which are specialized defensive cells. A synapomorphy is a characteristic that every species in the phylum shares. There are actually three types of cnidocytes: spirocysts, which shoot entangling threads, tichocysts, which shoot adhesive threads, and nematocysts, which shoot stinging threads. This video is going to go into the morphology of nematocysts and the mechanism behind how these tiny cells pack such a punch. We are going to be drawing the unfired nematocyst as well as a fired one. The motion is kind of like a torpedo, with the threads kept coiled up in the nematocyst cell until they are fired. Here is microscopic video footage of nematocysts firing in a jellyfish. The features we are going to be labeling include the cnidocyte cell, the nematocyst itself, the hinged operculum covering the unfired cell, the nematocyst tube, the cnidocyl, which acts as a trigger, the threads connecting to the barbed head, and the stylet, which is part of the barb. Now that we know more about the features, let's draw a cnidocyte cell. The interesting feature about the unfired nematocyst is that the barb is actually inverted inside of the nematocyst tube. When drawing the fired nematocyst, many of the outer features are the same. However, the hinged operculum is open, and the barb along with the connecting threads are ejected into the unfortunate victim, leaving the nematocyst cell empty. Thanks for joining us, and we hope that you can tune in for the next episode of Ocean Inverts.